Hello everyone, and welcome to episode 101. Today's show comes right on the heels of our special episode, episode 100, which was all about the Ediacaran biota. Now, if you haven't listened to that supersized special episode yet, you definitely should. It's really awesome, and it covered the early evolution of the metazoans, including, among many other strange and remarkable creatures, several groups that are still alive today, like the sponges and the Cnidarians. The Cnidarians, in particular, are some of the strangest animals in the world's oceans, representing one of the oldest lineages of animal life still alive today, younger only than the ctenophores and the sponges themselves. Today's show will be all about these Cnidarians, including the charismatic jellyfish and their Hydra, sea anemone, and coral relatives. Cnidaria is a relatively diverse clade with more than 11,000 known species. The majority of these, well over 6,000 of them, are corals and sea anemones and other sessile seafloor-dwelling animals. These represent some of the most basal Cnidarian groups. They're the descendants of the earliest offshoots of these ancient forebears and their titular cnidocytes, those infamous stinging cells that are shared among all of their descendants. In the fossil record, the oldest unambiguous Cnidarians are the Aurora lumina and the Hautia, both of them sessile, sea anemone-like organisms from about 560 million years ago. Another large portion of the Cnidarians, more than 3,500 species, belong to a group called the Hydrozoa. These amazing and extremely weird creatures include the freshwater Hydra, which is functionally immortal, and the beautiful array of Siphonophores, which are ghostly, translucent, alien-like colonies of Hydrozoans. They appear to be singular creatures, but they are in fact conglomerations of multiple smaller individuals all working together. Recent genetic analysis has identified a few smaller niche groups within Cnidaria, including the Staurozoa, the Polypodiazoa, and the Myxozoa. We'll take a look at all of these weirdos and their neat lifestyles later on in the episode. But for now, to round out the gallery of Cnidarian biodiversity, we come to perhaps their most charismatic and well-known representatives, the box jellies, which emerged in the mid-Cambrian, and the true jellyfish, which emerged a little bit earlier, probably in the late Ediacaran, somewhere between 540 and 560 million years ago. Surprisingly, these two most well-known Cnidarian groups are actually among the smallest and least diverse. There's fewer than 230 known species of jellyfish, and at the time of this recording, there's only 42 known species of box jellies. But despite their relatively low biodiversity compared to other Cnidarian groups, they are widespread and abundant. Jellyfish colonies can contain hundreds of thousands, or even millions, if not billions, of rapidly reproducing individuals. To the surprise of many scientists, recent studies have found that all of this raw jellyfish biomass is actually a really important food source for many animals. Despite each individual jellyfish being mostly water and having very few calories, they're easy to catch and they're easy to digest, and they play a counterintuitively important role in the aquatic food web. In today's episode, we'll look at the anatomy, life cycle, and ecology of every one of these groups. We'll explore how the Cnidarians live their lives, how they cooperate and compete amongst themselves, and how they affect the greater aquatic ecosystem as a whole. With that said, let's begin our story 580 million years ago, in the late heart of the Ediacaran. In this early era of complex life, the food webs and ecosystems were much simpler. In the shallow water areas, all kinds of algae made slick the rocks and seafloor where sunlight still reached, and from them eventually came the earliest multicellular plants, like the thallophytes and tiny primitive seaweed. In deeper water, there were ancient aquatic fungi and frond-like rangiomorphs. The sponges passively filter-fed on detritus, like the exudates and debris from microbial colonies and algae, and the decaying bits and pieces of everything else. Pretty much all of these organisms were largely immobile. They were bound to the seafloor. So too were these early Cnidarians, these corals and sea anemones of the class Anthozoa. I explored a few of these in great detail in the previous episode, like the predatory Aurora lumina and the curiously muscled Hautia. In this early stage, these Cnidarians distinguished themselves from their spongy cousins in several very important ways. They had developed a second germ layer in their embryonic state, representing the dawn of the diploblasts and the evolutionary capacity to produce a wider variety of cell and tissue types. And two, by developing a true radial symmetry in the overall body plan. They had evolved a body with the shape of a column, or a round-sided block, often described as a cup or a bowl. Each germ layer in the diploblast embryo will develop into a thin epithelial layer. These are the outer layer of the body and the inner lining of the gut orifice. These epithelial layers are thin enough that typical respiratory functions, like the exchange of oxygen and carbon dioxide, can be accomplished through simple diffusion, and it doesn't require any specialized respiratory organs. These epithelial layers also secrete fibrous proteins along their inner surface, forming a basement membrane. These basement membranes are analogous to the cement foundation of a building. It's not used for occupancy or day-to-day activities. Its purpose is purely structural. The basement membranes are essentially tough sheets of material that strengthen the body and the tissues, allowing it to endure greater pressures and forces. These epithelial layers are initially just a single layer of cells thick, but as the tissue develops and differentiates, the mature epithelia is usually several layers of intertwined and overlapping cells of various specializations. For example, a curious kind of muscle cell is incorporated into the epithelial layer, forming rows of protein-enriched striations that allow the entire body to flex and contract. Other types of cells in the epidermis exist in close association with these rows of muscle cells, like the sensory and motor cells in a basic neural system, which are the circuitry that controls and coordinates muscle activity in response to the sensed world around it. The sensory cells are a noted improvement on the pseudoneural sensory cells seen in the sponges, as the Cnidarian neural cells use true synapses and a body-wide net or network to optimize the coordination of muscle contractions. To expand a bit on this topic of the Cnidarian nervous system, they don't have a brain. They don't have a centralized nervous system at all. It's a very decentralized system, which in most species manifests as simple partnerships between proximal sensory and motor nerve cells embedded in this epithelial layer surrounding the body. These sensory and motor nerve cells communicate with other proximal cells, sometimes forming small ganglia to control local behaviors, whereas body-wide behaviors involve the recruitment of mini nerve cells, or as many ganglia as possible. Many species have developed a decentralized but coherent nerve net, which is a loose reticulating basket of nerve cells woven into the epithelial structure. This nerve net connects the sensory and motor neurons across the body. It enables a greater degree of cooperation and synchronization. That translates at the macro scale to more sophisticated and effective behaviors. These neurons communicate by sending neurotransmitters across synapse-like gaps. The variety of neurotransmitters that they use is lower than that seen in vertebrates, but the neurotransmitters that they do use include the excitatory glutamate, the inhibitory GABA, and other regulatory neurotransmitters like acetylcholine. I think it's pretty incredible that these ancient organisms use a similar set of neurotransmitters to serve similar functions as compared to the more derived triploblast protostomes and deuterostomes. It's a subtle but powerful testament to our shared collective ancestry. The nervous system sensory cells can detect things like temperature or pressure or certain chemicals, and they feed signals to motor neurons that adjust the behavior of the cnidarian so that it can respond to its environment in appropriate ways. For example, in jellyfish and other cnidarians with the free-swimming Medusa body form, and I'll get into more detail on the body forms in a few minutes, they've evolved statocysts to help them determine their orientation. Statocysts are cells that contain internal chambers lined with cilia-like hairs. Within the chamber sits a clump of mineralized crystals, which are heavier than water. As the jellyfish swims around, the crystals will fall downward because of gravity, and the location of the crystal can be detected by those cilia-like hairs. This informs the statocyst which way is down, and the signal is relayed through the neural net. If the jellyfish is tilted in a weird direction, like at some oblique angle or something, the statocysts will detect this, and they'll signal to the appropriate motor neurons in this decentralized net to increase their activity, to flex the body to turn it upright. Perhaps the most surprising and, dare I say, unsettling neural adaptation in the cnidarians are the development of eyes. Many species of Medusazoan have simple eyes, simple ocelli, which are little more than clusters of heavily pigmented cells, whose pigments are photoreceptive and can respond to light. These simple eyes, these ocelli, cannot focus, they can't make out details, and they certainly can't form images. But what they can do is detect the direction of the sun or the full moon, and they can detect the shadows of things moving between them and a light source. This rudimentary form of sight enabled the Medusazoans to better navigate around obstacles and integrate the light pattern of the day-night cycle into their life strategies. Within the cnidarian clade, eyes have been evolved independently no less than 8 times. However, this has only happened among the Medusazoans, like the Hydra and the jellyfish. No anthozoans, so no stationary cnidarian with a polyp body form, like a coral or a sea anemone, has ever developed eyes. And why would they? They can't move, they can't follow the sun or run away from predators. There's no point to having eyes, it's a waste of resources. But for the free-swimming Medusa, which can move around to some degree under their own agency, they derive great benefit from being able to see what they're swimming next to. Or, alternatively, being able to see what's swimming next to them. Although I should note that many species of jellyfish have been experimentally tested to see how they respond to light, and lots of them don't. They have ocelli, but varying levels of light intensity or movement of the light source don't seem to elicit significant behavioral changes. So, in at least some species, there's a question of whether the ocelli are even functional or not. If they are functional, then there's a question of whether this sensory signal means anything to the jellyfish, whether it's even being relayed to the rest of the neural net and being integrated into the rest of the calculations being made about how to move the body and how to respond to environmental stimuli. We really don't quite know what's going on here, but of course, research is ongoing. Now, on the other hand, there are several clades of box jellies with extremely sophisticated eyes, which definitely confer visual abilities and, subsequently, complex and informed behaviors. In the box jellies, the eyes have been developed into a fully fledged, complex eye with a 3-layered retina, a cornea, and a lens made of a unique crystalline compound. These can not only sense the direction and the intensity of light, but they can actually form images and produce complex visual data. The shape of this complex eye varies across the box jellies, including circular and slit-shaped eyes, which could mean that these complex eyes are either homologous features that have been altered in cousin lineages, or they could all be independently derived forms of complex eye. One thing for certain, though, is that Seeing a box jellyfish with 4 uncannily human-like eyeballs mounted in its fluffy epithelia is beyond creepy. You really see this a lot in the Trepidalia genus, where these small, fleshy eyes with irises and pupils and everything are nested at regular intervals around the rim of the body, of the bell or cup shape of the box jelly. It really makes you wonder if these eyes can form images and these Cnidarians can see you when you're like, say you're scuba diving or something and you're up close to one and you're looking at it, you're looking the box jelly right in its eye. What is it seeing? What does it think about what it's seeing? Does it have an experience like that? Is there something that it feels like to be a box jellyfish with 360 degrees of vision conferred by 4 weird eyeballs that ring your body? What is that like? This is an idea that harkens back to the hard problem of consciousness. I mean, basically it is the hard problem of consciousness. Really, the thing that you have to ask here, beyond just the eyeballs themselves and the visual sense, is what is it like to be an organism with a decentralized nervous system? What is it like to exist and sense the world and have some sort of identity, if you can even call it that, some sort of awareness in a body that has a decentralized nervous system, as opposed to like a centralized system with a spine and a brain? It's really difficult to even imagine that. And you have to wonder that it would be kind of mind-blowing if, say, in the future we research this and we find out that, somehow we find out that the experience really isn't all that different from having a centralized nervous system. I mean, that would throw everybody for a loop. Okay, but I'm really getting into the weeds here, so let's get back on the trail. Coming back to the embryo and Cnidarian development, the embryonic epithelial layers will also secrete, in addition to that basement membrane, they'll also secrete a looser mixture of connective fibers in the interstitial space, so that space between them, between the gut lining and the outer layer of the body, to form a jelly-like mesoglia. The mesoglia fills in the internal void between the epidermis and the gut lining, effectively becoming a hydrostatic skeleton. Many species of coral, sea anemone, and jellyfish also have muscle cells that either reach into or exist entirely within the mesoglia. Alongside them are a group of rather unassuming janitor cells that help to maintain the mesoglia and do basic repairs and cleaning functions. These are somewhat like amoeba in their shape and movement style, and they seem homologous, albeit in a reduced form, of the rich community of amoeba-like cells that exists within the mesohyl of sponges. Earlier in the episode, when I was talking about the anthozoan body form, I described it as cup or bowl-shaped. Well, along the upper edges of this cylindrical body form, analogous to the open rim of the cup or the bowl, these had evolved a set of tentacles. Reaching lazily upward into the water column, the tentacles were an adaptation that enabled predation, as seen in the first known animal predator, the Ediacaran sea anemone Aurora lumina. Their tentacles were weaponized with yet another evolutionary innovation of the Cnidarians, perhaps their most distinguishing feature, the cnidocytes, or the stinging cells. Now there's actually quite a few different types of cnidocytes, although they all share the same general format. All cnidocytes are specialized cells that can shoot out some sort of molecular weapon to disable or harm a prey animal. Some of these molecular weapons are sticky pads, sort of like the colloblasts seen in the ctenophores. They shoot out, stick to the target prey animal, and help to constrain it or entangle it. Another type of cnidocyte shoots out a fibrous thread with a loop at the end, like a lasso or a noose, which can snag onto some protruding feature on the prey animal and trap it. The most common cnidocyte, and by far the most dangerous, are the penetrating type, that shoots a molecular harpoon loaded with venoms. The cnidocyte is a narrow, bulb-shaped cell containing a hollow, tube-like piece of toxin-loaded ordnance. A thin hair extends outwards from the cell, and when this hair is mechanically or chemically triggered, like when it brushes against the scaly body of a passing fish, that initiates the firing process. There's a capsule of calcium ions kept on standby within the cnidocyte, and when the firing process begins, when that sensory hair is triggered, these calcium ions are rapidly released into the cytoplasm. The water potential of the cytoplasm suddenly drops as it's flooded with solutes, and osmosis leads to water surging into the cell. It is this sudden influx of water caused by a surge in the solutes that squeezes out the tubule and leads to its forceful ejection. The tube-like structure is actually an inverted tip of the bulb, so it's sort of like the shape of a radish, except if the thin central root of the radish was turned inside out and coiled up inside the radish itself. When water rushes in, it everts the tube section, or the radish root, metaphorically. This change in water potential and the surge of water turns it right side out with tremendous speed. From initiation of the firing process with the activation of the trigger hair to the surge in water that makes the whole thing whip out and sting at the target prey animal, the whole process takes about 2 to 7 milliseconds. This microscopic firing process has been calculated to accelerate the tube-shaped harpoon at tens of thousands to millions of G, which is more than enough for the harpoon to penetrate the flesh of the prey animal. If we calculate the acceleration by the mass and the, albeit very tiny surface area of the harpoon's tip, the resulting force is nothing less than astonishing. A single cnidocyte, a single cell, can strike a target with 7 gigapascals of pressure at the harpoon's tip, which is comparable to actual bullets fired out of a modern firearm. Of course, this pressure is highly localized at the nanometer scale, which is why it doesn't blow apart the prey animal into tiny wet pieces like an actual bullet would. Just like the structural specifics of the cnidocyte, the venom itself that gets injected into the prey animal varies greatly from cnidarian to cnidarian. In many cases, the venom is optimized for a particular prey animal. The defenses of the prey animal also play a big role. For example, many sea anemones have cnidocytes that can paralyze or kill small aquatic invertebrates, but they do nothing to humans because they're just too weak to penetrate our skin. Meanwhile, the box jellies of the class Cubozoa have cnidocytes that are strong enough to sting and even kill an adult human. There's many cnidarians whose stings are notoriously and excruciatingly painful, and sometimes even fatal for humans, like the Portuguese man-o'-war, Phasalia phasalis. Perhaps the most classic example is the orange and white clownfish, Amphiprion percula, which is thought to have co-evolved with its sea anemone host, usually Heteractis magnifica, so that the sea anemone's venoms don't hurt it. However, clownfish can be hurt and killed by their own sea anemones if the clownfish's mucus coating is removed, so this suggests that the mucus coating offers some sort of protective benefit. A leading idea here is that the mucus, being largely made up of carbohydrates instead of proteins, is able to evade detection by the chemosensory trigger mechanism. The trigger mechanism needs to be activated by the dynamic electrochemical environment of a protein, whose side chains might have positively or negatively charged polar groups or something like that. Whereas carbohydrates tend to be much less polarized. They might have a few oxygen atoms embedded in there here and there, and that creates a little bit of electronegativity, but for the most part, they're carbons and hydrogens, so there's not a very polarized or dynamic electrochemical environment. There's nothing exciting like that would trigger this chemosensory mechanism and induce the sea anemone to sting the clownfish. Okay, so I've been talking about the many cells in the cnidarian body plan, from their muscle cells to their stingers. This body plan, as I described, is radially symmetrical and cylindrical in shape, like a cup or a bowl. The cnidarians manifest this cylindrical form in one of 2 ways, as a polyp or as a medusa. The polyp body form is oriented mouth up, with its base cemented to the substrate, and the medusa body form is oriented mouth down, with the creature moving freely through the water, able to swim with its own muscular contractions. Some cnidarians will stay in the polyp form for their entire life, while others will transition into the medusa stage as part of their life cycle. The polyp body form is generally stationary, growing on the substrate like a stubby arm with venomous fingers. The inner core of the polyp is a hollow gut chamber that's lined with fluffy folds of absorptive gastrodermal tissue. The upper portion of the body is turgid with the jelly-like mesoglea, the internal pressure erecting the many stinging tentacles, reaching up and sieving through the water for food. The base of the polyp is mounted to a substrate, but what this substrate is generally varies depending on the species and the location of the habitat. Some of these polyps that live in colonies may have cenosarc tissue connecting them to nearby polyps and creating this larger, continuous fleshy mass. Some of them may share a deposited foundational layer of exoskeletal secretions, like for example in some branching soft corals, where the fleshy soft skin of the colony may be growing around a horny endoskeleton, a mineralized core that runs through them like a bone in a limb. The stony corals form calcium carbonate skeletons, which are deposited around the base of the polyp, sort of like a mineralized pair of pants. Each polyp forms a little socket that it lives in, called a corallite. Most species of polyp live in groups or colonies connected by that cenosarc tissue I described. Corals, for example, may appear like clusters of cones or lumps or pillars. And each protuberance here is a single individual polyp, enclosing a fleshy internal body and a cluster of soft tentacles that are released at night to grasp out into the water in a blind attempt to catch food. All of the polyps in a colony are genetically identical, and they grow from a shared layer of vascularized tissue. This allows them to distribute nutrients among one another and ensure that everyone is equally well fed. Fundamentally, this is the purpose of a community. You work together to help keep each other alive, and the coral do a pretty good job at it. In the stony corals, their skeletal secretions form a strong mineral base that the colony grows from. It's actually strong enough to withstand tidal forces if need be. For example, the Acropa cervicornis staghorn coral lives in shallow water in and around the Caribbean Sea. It's exposed to the regular beating of waves and the shifts in the tide, so it has taken on a highly branching form with long, thin arms. Each arm produces little resistance to the current, as most water just easily flows around it, so the staghorn coral can grow to relatively large sizes without fear of breaking off entire limbs. In contrast, if you go to deeper water where the current isn't necessarily as strong and as physically destructive, you might see forms of corals like the pillar coral Dendrogyra cylindris, which makes massive pointed columns of red flesh, sort of like a baobab tree but without the leaves on top, and with beefy thorns sticking out along the sides where the actual coral polyps reside. Many of the tree-like forms that you see in a reef environment are also corals, with the most visually recognizable being creatures like table coral, the Gorgonian sea fans, and sea whips, and if we expand out a little bit more, the unique and often enormous brain corals, whose surface is covered in rows of soft ridges and sulci, like a giant, super-intelligent aquatic brain. The sea anemones are larger polyps that usually live independently and not in groups. They're also significantly larger than an individual polyp in a colony species, with much more complex individual bodies, many more tentacles, and ultimately an entirely different ecological role than the coral polyps that live in colonies. The many forms that sea anemones take are all truly astounding in their diversity and beauty. Some species have just a few thick tentacles, while others have many thin ones. Some are long and wiry, while some are short and stubby. Some raise their tentacles upwards, while others spread them out in all directions. Often, the rim and mouth of the sea anemone may be like a wreath, with short, bristly, or fuzzy-looking tentacles that are kept close to the body. The dangerous Hell's Fire sea anemone, Actinodendron arboreum, looks like a tree, or perhaps more like an aquatic cauliflower. Some of the largest include the carpet sea anemones, which dwell in high-density tropical coral reefs, where they form a disc or a plate that grows up to a meter wide, with an upper surface lined with thousands of small tentacles. This disc body form can flex and bend to reach up and tag a prey animal, slapping it and paralyzing it and catching it as it drifts down, to then slowly push its paralyzed form towards the central mouth orifice to be ingested. There's usually cnidocytes lining the inner gut tissue as well, so that the prey animal is continually stung as it's being digested. This is to ensure that at no point in the process, particularly earlier on in the process, it doesn't suddenly recover, it doesn't wake up and fight back, which would quite likely be lethal for the sea anemone. In many of these carpet sea anemones, they develop a greenish tint from all of the algae that grow symbiotically in the giant fleshy disc. These algae will photosynthesize and share their sugar with the coral. Many sea anemones, in fact, integrate algae into their skin and tentacles. The algae enjoy physical protection from herbivores and microbial parasites, thanks in part to the stinging cells keeping them away, and the sea anemone, of course, benefits from having some extra nutrition, the excess sugar that the algae is willing to share. As we already talked about with the clownfish, sea anemones can also form symbioses with larger animals like crustaceans or fish. The clownfish is the classic example, and they're able to resist stings, probably due to that special sugar-based slime on their scales. But other symbioses with sea anemones and other animals are really interesting and worth a note. The small Bengai cardinalfish, Pteropogon carderni, found in the waters of Indonesia, has a life cycle spent almost entirely among corals and anemones. Their life cycle here, where they'll shed scales and bring back food, will inevitably create detritus, biological debris, within this sea anemone habitat, the sea anemone home, if you will. And of course, the sea anemone benefits because this is all food that it can scoop up and eat. Outside Indonesia, from East Africa up to southern Japan, the domino damsel Dascylus trimaculatus plays a similar ecological role. Many crustaceans also form similar ecological relationships with sea anemones. And then you have things like hermit crabs, which make a really odd case, with a symbiosis that kind of mirrors their use of sponges. You may remember from the sponge episode that hermit crabs carry sponges along and use them as shields or camouflage, and they transfer them from shell to shell. So as the hermit crab grows and it moves into a bigger shell, it'll bring its little sponge buddy along. Well, hermit crabs will also do something very similar with small sea anemones, putting them on the shell and taking advantage of their stingers and tentacles to use them as a kind of mobile air defense and a simultaneous food capturing service. And then you have the boxer shrimp, which are already crazy on their own, but these have been known to clip off bits of anemone with the tentacles attached, and then paste them onto their claws to use them like venomous, stinging boxing gloves, to put just that much more punch in their attacks against fish and other crustaceans. A major evolutionary innovation among the sea anemones was the ability to construct a vertically oriented burrow in the substrate, to line it with fleshy secretions until it's kind of like a little blanketed pocket or a sheath, and then they can use that as a homemade tunnel. The sea anemone can retract into the sheath or the tunnel if they want to hide from predators, or if they want to set up an ambush for an oncoming prey animal, or if they're just tired and seeking to conserve energy in safety. They retract back into this sheath and hide. Other sea anemones enjoy a curious lifestyle where they're mostly immobile and mostly stuck in one place, except for when they're not. Yeah, so I never knew this. I always thought sea anemones were stuck where they were for their entire life. But no, that's not true. They don't produce rhizoid-like tissue that roots them to the substrate, like you'd see in a sponge or something. But instead, they produce a foot, or a pad, sort of like what you'd see on a slug or a snail. And they can use this to slowly, very slowly, inch around and explore their habitat. They'll generally stay in one spot for weeks or months at a time, and then they'll move somewhere else and settle down for another period of immobility. They generally migrate to find better water and better feeding areas. It's just that this search process and this migration to the area is incredibly slow. They move at a glacial pace across the ground, although a small selection of species are able to push off, so to speak, and float in the current for short distances. They do this by rudimentary swimming motions in their base, or regular beating of the tentacles, sort of like giant cilia, or even by engorging themselves with water and then being rolled about like a beach ball in the current, as seen in the sea onion sea anemone Paranthus rapiformis. None of this is true swimming per se, as it can't be sustained indefinitely, and they will quickly come back down and establish themselves on the ground again. So in essence, that is the polyp body form. The other Cnidarian body form turns this cup or bowl shape upside down. It's more of a bell shape. This is the Medusa body form, and it's essentially the free-moving, unbound version of the Cnidarian body plan. Its mesoglia is thicker and much more firm than the mesoglia in the stationary, often mineralized polyps. The Medusa, in contrast, is not immobile and stuck to the seafloor. It can pulsate with muscular contractions and swim under its own power through the water. Where the polyp is typically cylindrical or conical, the Medusa body form is more hydrodynamic. The top part of the body is rounded like a dome. The edges curl down to partially enclose the mouth, which is now at the center of the bottom of the body. This enclosed space within the bell, surrounding the mouth and the underbelly of the Medusa's body, contains a small quantity of water. When the thin, one-cell thick sheet of muscle tissue in the body wall contracts, that water is forcefully expelled out of the enclosed space, which then gently propels the Medusa in one direction or another. Now the tissue isn't very strong, so their ability to swim against the current is very limited. And furthermore, there's no counter muscles to pull back the body wall, but rather, the mesoglia is so firm and rubbery, and the muscles are generally so soft and relatively weak, that this can just spring the body form back to its original shape, naturally relaxing and expanding to let in more water for the next contraction. A ring of tentacles grows out from the rim of the bell shape, trailing behind the Medusa zone in the water as it swims along. These tentacles can be twitched and flicked and numbly dragged around to sweep up prey animals, which can then be stung and disabled. Many Medusazoans also have another feature. In addition to their tentacles, they may have thin appendages protruding from the mouth tissue. These may look like weird tentacles, as they're often frilled and ruffled, or they have branching tendrils, but they are, in fact, a special type of limb called an oral arm. These oral arms can filter food particles in the water, or entangle a poor paralyzed prey animal, and then wipe themselves off on the mouth. Any food they catch is scraped off and ingested. In many species, like the northern sea nettle Chrysora melanaster, the oral arms are significantly longer than any of the actual tentacles, trailing behind a rounded cap that can be up to 60 centimeters in diameter. Inside the gut, digestive enzymes are released, and the prey biomass is broken down into a pulpy soup. The prey animal, now in liquid form, is dissolved into the watery internal fluids and spread throughout the Cnidarian's body. Nutrients are picked up by the thin epidermal cells via diffusion, and if the species is, for example, a polyp living in a colony with vascularized tunnels in the cenosarc connecting them all together, then nutrients can be shared with all members of the colony. Within each polyp, or Medusa that has just digested a meal, there may be indigestible portions left over, like hard parts or durable globs of connective tissue that couldn't be broken down. These will be expelled out through the mouth, along with a flush of wastewater containing the ammonia byproducts of Cnidarian metabolism. Now the Cnidarians with the Medusa body form, like the jellyfish, are typically passive hunters. They try to direct themselves towards sources of food, like areas with fish or high concentrations of zooplankton. They spread out their tentacles, expanding themselves like an opening net to scoop up and catch food. The simple act of swimming also brings zooplankton closer to the mouth, because after every jet stream of water that gets pumped out, new water comes rushing in as the body expands, and this can be sieved and tasted for food particles before being pumped out with the next contraction. Jellyfish and box jellies can also catch prey by dragging their tentacles behind them or near the seafloor, where they can entangle crustaceans, paralyze fish, and sweep up tiny larvae and fish eggs and other such things. A huge part of a jellyfish's diet is, perhaps surprisingly, other jellyfish. After all, jellyfish come in a wide variety of shapes and sizes, so some can easily engulf and consume others. And pretty much the only thing that's slower than a jellyfish is another jellyfish. When almost everything else can outrun you, even a crab, those things that can't outrun you will inevitably end up being a bigger part of your diet. And those things tend to be other, smaller, slower jellyfish. If we step back a bit and look at all of the jellyfish, their slowness and their general inability to quickly fight back makes them an easy snack for larger predators, like sharks and turtles and various ray-finned fish. But they're not very calorie-dense, and they aren't a primary food source for anything, like penguins or tuna or something. It's usually just their convenience that's a big selling point. In most areas, the population of large medusazoans, like mature jellyfish, is kept in check by the occasional grazing of 1,000 bigger aquatic and semi-aquatic animals. But sometimes, jellyfish populations can get out of control, and this can have devastating consequences on the local ecology in both the short and long term. For example, there's events called jellyfish blooms, and they're the result of a confluence of factors all aligning in the jellyfish's favor. Consider, for example, that warmer water is generally less able to absorb oxygen than colder water, so tropical areas or warm water currents tend to be less well oxygenated than colder water areas. This can make life difficult for many marine animals, but jellyfish can endure and even thrive in warmer, low-oxygen water. Consider a related event called an algal bloom. Algae are often limited in their growth by a lack of certain mineral nutrients, like iron or copper. When it rains, runoff from dry land can carry sediment into the water, which brings in mineral particles. These particles will be dissolved into the water. Eventually, they'll precipitate out, but for a time, there's enough dissolved nutrients for the algae to go crazy. Their populations explode, and they appear on the surface of the water as vast discolored patches of microbial growth. The problem is that algal blooms use up a ton of biological resources. They use up a ton of oxygen, leaving behind anoxic dead zones. Fish and many aquatic invertebrates cannot tolerate these dead zones, so they either swim away or run away, or they suffocate and die. When the mineral sediment settles to the seafloor and the algal bloom dies off, this anoxic zone can linger for a long time until it slowly gets repopulated and reoxygenated. A problem here is that the jellyfish are much more comfortable in these dead zones than other organisms, like any given ray-finned fish. Jellyfish can thus colonize these areas first, and they hold all the cards. They're predators that are really good at gobbling up fish eggs, and if any fish are born from, any eggs that are lucky enough to survive, well, these young fish fry have to compete with the jellyfish for the same food, like plankton and crustacean larvae. In this way, the jellyfish can prevent any sizable fish populations from getting a foothold and recovering in the area, which can have dire ecological consequences, including overall drops in biodiversity and primary production. Fortunately for the jellyfish, but unfortunately for pretty much everything else, global climate change is creating warmer, saltier, lower oxygen oceans. Jellyfish are currently surging into new areas as invasive species, and they will presumably become an increasingly important part of marine ecosystems in the future. We must also face the fact that their increased presence will likely mean a global reduction in fish at all levels of the aquatic ecosystem, including the fish we harvest for food. Okay, so let's get to a topic that I've been really excited to get to: the Cnidarian life cycle. Of all the things I've read about across the entire animal kingdom, there is almost nothing as weird, as unusual, as freakishly alien as Cnidarian lifestyles and reproductive strategies. As with all things biological, there's a lot of variety at every level here, so let's look at a few common patterns. Consider the jellyfish and the box jellies, two groups of medusazoans that begin their lives as free-swimming larvae. These larvae are shaped like sausages or cones, with the thicker end forward and the mouth at the back. They're akin to small bags or discs with a vaguely bilateral symmetry, and they're lined with cilia, which they use to swim in the water. The larvae do not live independently for any significant time, but instead they quickly find a suitable place on the seafloor or the substrate to bind and attach to. Once anchored in place, the larva morphs into a cup-like polyp form. This will then grow into a sea anemone-like form, so basically a large polyp body form. They'll develop a cup shape with tentacles around the rim and everything. Then this cup shape will extend, growing taller and more column or pillar-like. At some point, the tentacles are absorbed and dissolved back into the main body, initiating a really cool process called strobilation. Upon the absorption of the tentacles, the body mass will now appear like a tall chimney. Suddenly, deep folds appear like bands or rings along the chimney. These transverse constrictions, as they're called, appear first near the tip of the body column, and as they deepen, more rings will appear and constrict further down the body. Eventually, the entire structure appears like a column of stacked discs, no more than a few millimeters wide. As the strobilation process completes, the discs will fully separate and break off, whereupon they grow their own tentacles and achieve the Medusa form. These Medusa are all clones of the parent, representing a kind of asexual reproduction. The polyp form can also reproduce asexually by budding or by fragmentation, either by splitting down a designed central line or by a piece being broken off and regenerating itself somewhere else. For example, some Cnidarians with the polyp body form can slowly move across the seafloor, and as they do, they'll break off pieces of the footpad and the associated section of the polyp's flesh. These left-behind pieces will regenerate into new individual clones. Although the Medusa body form is produced asexually, the larva, or the polyp that it grows from, is not. They're produced through a sexual process, so the clones that it produces are either male or female. After releasing these Medusa offspring, the polyp can persist and regenerate to undergo strobilation multiple times, producing countless more clones of one sex or the other. Now, most of these jellyfish and box jelly Medusa clones are either male or female, although some species are hermaphrodites. But in any case, the adult medusazoans produce eggs and sperm, with gonad tissue kept in the lining of their gut. The gametes are released into the gut and then expelled in the same way as wastewaters, with a muscular contraction. External fertilization in the jellyfish requires huge quantities of gametes, and the jellyfish leaned in hard to this evolutionary pressure. The sheer volume of gamete secretions during a mating season can visibly discolor the water. When sperm meets egg and fertilization is achieved, the embryo forms a blastula, which then matures into the larval form. The larva then seeks a place to settle on the substrate, and the process begins again. As this is a messy biological process, there's a lot of variation at every step in the process. The anthozoans, for example, the corals and sea anemones, obviously don't have a Medusa life stage. Instead of strobilation, the mature polyp form simply secretes gametes, and external fertilization creates larvae to then create new polyps, and they just grow big and then do the whole process over again. So it's a bit simpler in that regard. But the process varies a lot, even among jellyfish. Depending on the species, the polyp form may develop individually, or it may blossom into a colony. The polyp may not even be settled on the substrate. It could be free-floating, or bound to a piece of flotsam, or even a planktonic animal. Keep in mind that these things are usually very small, on the order of micrometers to maybe even millimeters. The time spent in all of these stages varies wildly too. Some species produce a yolk for their larvae to feed off of, which allows the larva to stay alive in the larval form for weeks, up to 80, 90, or even 100 days before they settle to the seafloor. The polyp stage can also last for years, and for those that strobilate and regenerate, they may be able to survive for possibly over a decade or more, producing new clones every year. There's even a jellyfish species called Turritopsis dohrnii, which is known as the immortal jellyfish because of its ability to revert from Medusa to polyp body form. This process can be triggered when the Medusa is injured, or when it's exposed to sudden environmental changes in salinity or temperature, or if it risks starving to death. In any of these challenging moments, the Medusa of Turritopsis dohrnii begins to dissolve its mesoglea and collapse the bell shape of its body. The tentacles are broken down and the nubs are absorbed. Most individuals will then settle to the seafloor in a dormant state, forming a pocket or a cyst-like structure on the ground. Eventually, this cyst will bloom new polyps. These will grow and branch, sometimes into a hydroid form, and then finally they'll regenerate a new Medusa adult form. In effect, the Turritopsis dohrnii can revert itself back to its Cnidarian infancy, and then grow through its life stages like normal, again and again, basically indefinitely. This ability makes it functionally immortal, so as long as it's not destroyed by a predator or succumbs to any adverse environmental changes, an individual Turritopsis dohrnii jellyfish could live, theoretically, forever. I mentioned a word there a minute ago that may have given you pause. I mentioned the hydroid form that might occasionally be seen by the immortal jellyfish as it regenerates and goes through its life cycle again. This hydroid form is a very curious life stage in some Cnidarians, most of them belonging to the class Hydrozoa. In this hydroid life stage, multiple polyps live together in a colony, which is usually anchored to the ground. It can have the appearance of weird animal seaweed, like the hydroid form of Aglaophenia cupricina. In other cases, the hydroid form appears like a medieval mace, or like a weird sea anemone with a long stalk and knobbly oral region, with tentacles protruding in all directions. The reason that I wanted to mention the hydroid body form was to introduce the class Hydrozoa. The Hydrozoa are particularly strange Cnidarians that usually either live entirely as a polyp or entirely as a Medusa. Of those species that produce both body forms, the Medusa is often a tiny blob kept near or on the parent polyp. In many of these species, it's not even detached, and it's been so reduced that it doesn't even have any internal structures or a mouth. Like many species of male anglerfish, some of the Hydrozoans' Medusa forms are little more than a glorified gonad glued to the side of the parent or of the female. Within the class Hydrozoa, there is the order Siphonophores. This is a really awesome group that I wanted to mention, because the Siphonophores often have a free-swimming Medusa form, but it's not a single individual. It's actually a colony structure made up of multiple, often specialized polyps all working together. Sort of like a larva, there's an initial zooid that reproduces asexually to create a colony of polyp-like zooids, except instead of being identical clones, these cloned zooids will undergo post-embryonic mutations. Various zooids in the colony will be modified in this way or that way, creating diverse subunits with specialized abilities. The zooids are then arranged along a central stem, which gives them a kind of bilateral symmetry that's not seen in other cnidarians. The particular arrangement of zooids along the stem depends on their specialization, on their function, and how they operate within the colony as a whole. Specialized zooids can also replicate themselves via budding. For example, the zooids within a siphonophore can be in a polyp or Medusa form, and each form can be specialized for certain functions. For example, the nectophores are Medusa-form zooids that operate like water jets. They help shoot out water and thus propel the siphonophore in a desired direction. Conversely, pneumatophores are structures on the original zooid that form giant gas bladders. These air-filled sacs are used for buoyancy, to maintain an upright orientation, and possibly to gauge water pressure. Zooids involved in processing food turn into gastrozooids and palpons, and the gonophores are zooids that are specialized for producing gametes and helping reproduction. Siphonophores are grouped together kind of like plants, in the sense that they're either monoecious, where a single colony can have male and female gonophores, or dioecious, where a single colony, a single siphonophore, is either entirely male or entirely female. The siphonophores hunt in a manner similar to jellyfish. The gastrozooids are typically equipped with stinging tentacles, which they use to attack their prey during an ambush. To conserve energy, the siphonophore will typically float along quietly, ponderously, sometimes swimming cautiously, only reacting when something swims too close to it. The tentacles will be thrust out to wrap around the prey animal, and millions of cnidocytes will shoot their venomous harpoon stingers at it. The venoms of the siphonophores are neurotoxic and often very dangerous, if not outright deadly, to prey animals. Now, part of their attack strategy involves bioluminescence, where flashes of light are produced along the tentacles in a way that confuses the prey animal and prevents it from figuring out what's attacking it, or how to fight back, or where to escape. These bioluminescent compounds are produced in tentilla along the tentacles, and they can also be used like an anglerfish's bauble to soothe and lure in prey animals, like fish. Certain techniques, like using a red light with the occasional flick, can simulate the movements of plankton and draw in hungry fish or crabs or squid looking for a meal. Blue light can attract krill, and this can help the siphonophores compete with larger, faster-moving predators also seeking out the tiny food animals. Perhaps one of the most charismatic and well-known siphonophores is the Portuguese man-o'-war. It's an incredibly unique creature, with an engorged, translucent, asymmetrical pneumatophore that's even equipped with a sail to help it catch the wind at the water's surface and be pushed along. The asymmetrical, thumb-like shape of the pneumatophore can manifest as both left and right-handed versions. And this is actually really important, because the Portuguese man-o'-war cannot swim, and the handedness of the sail determines how it gets pushed by the wind, and this in turn can split up populations of these Portuguese man-o'-wars into left and right halves, each of them being pushed by the wind in different directions. This feature is thought to have evolved in order to protect the population from all being swept up on the shore or into some dangerous area where they're all destroyed. In this way, by having the left and right-handedness and the divergence in the way the wind pushes them, in this way at least some of them can survive and spread out somewhere else. Hanging under the pneumatophore are bundles of specialized zooids, including dactylozooids, also known as tentacular palpons, which are used for feeding, and of course the previously mentioned hanging tentacles of the gastrozooids. When a prey animal is paralyzed and captured, it's reeled in and destroyed by the various feeding appendages of the gastrozooids and the palpons. The siphonophores are prolific consumers of fish fry, of shrimp, crab, eggs, and of course, zooplankton. Their venom is extremely powerful, and painful too. It's able to rapidly paralyze and kill small to medium-sized fish, which substantially expands their menu of available prey. That's one of the benefits of investing so much energy in making potent neurotoxins. You can disable bigger prey animals and get bigger meals. So the Hydrozoans are really weird, even among the Cnidarians, which are already a really weird clade. There's several other really strange Cnidarian groups, several obscure offshoots that I wanted to briefly explore in today's episode. For example, there's an entire class of Medusazoan with about 50 species, called the Staurozoa, or the stalked jellyfish. They're sort of like the Cnidarian version of a crinoid, with a small, thin stalk connecting to the ground substrate. The main body is like a flipped-over starfish with 8 arms, and at the end of each arm there's a cluster of short tentacles that appear like a puffball, or pom-poms. The Staurozoans are small, no more than 1.5 inches tall. They dwell close to the seafloor in colder areas, like polar and boreal water, and in shallow water coastal areas too. Here, living among the rocks and the sand, they swipe plankton out of the water, and they clobber small crabs and shrimp. There's a neat little subgroup of anthozoans closely related to the soft corals called the Pennaculacea, or the sea pens. The name, sea pen, is a reference to a feather quill, as several species look like fleshy red quills sticking out of the sand. Most sea pens, however, are like tall flowers, or like medieval maces. Some of them even look like weird alternating ladders, with each step or frill or branch coated in tentacles that filter plankton out of the water. Like other corals, they're composed of a colony of polyps, but like the Hydrozoans, the polyps are not perfect clones, but instead they undergo post-embryonic mutations to develop specialized features so that they can perform specific roles. For example, one polyp will grow to form a long, narrow stalk. This is the stalk that connects the colony to the substrate, usually with a thickened footpad or anchor called a peduncle. The peduncle is somewhat like the fleshy sheath used by certain sea anemones. Some of the sea pens can collapse their entire body and retract it into the peduncle, which may be partially buried. Generally, larger sea pens will inhabit deeper water, which is more stable and less prone to the currents and pressures of the waves on the surface. This can uproot them, so to speak, causing them to lose out on a good feeding area and potentially injuring them as the current throws them around and scrapes them on rocks and other coral. Anyway, other polyps in the sea pen colony will specialize to be optimized for stinging, or for jet propulsion, or for gamete production, much like how the Hydrozoans do it. Now, if you thought all of these creatures were really strange, even creepy, well, there's even more creepy creatures among the Cnidaria. Consider the creepy, freaky, and quite lonely Polypodiazoa. Within this class, there's a single order, Polypodiidea, and within this order, there's a single family, Polypodiidae. This Polypodiidae family contains a single genus, Polypodium, and within that is the one lone species in the entire class, Polypodium hydriforme. Imagine a small jellyfish-like creature with plump tentacles armed with cnidocytes and ghostly pale, yellow-white, semi-translucent flesh. These guys are parasites of sturgeon and similar fish, and they're unique in several ways. As far as animals go, they're small. Really small. They're so small, in fact, that they live and grow inside the cells of their host. They spend part of their life cycle inside the eggs of various sturgeon, and at least one species of paddlefish. Here's how it works. In the winter and the spring, there are free-living adult Polypodium hydriforme populations living in the open. They reproduce asexually through a kind of organized fragmentation called paratomy. In the middle of the summer, when temperatures have risen and there's more daylight, they develop fragile pseudosexual organs in their epidermal tissue. These take the form of dense clusters of cells that form a gonad, and thin directional folds of tissue around it. Strangely, their reproductive strategy seems to be evolving away from sexual replication, and more towards a kind of permanent asexual replication. It's really strange, because the females, or at least the individuals that produce ovaries, are the ones that don't reproduce. Their gonads seem to produce inactive diploid cells that don't undergo meiosis. In other words, they don't produce functional eggs. In contrast, the testes will undergo 2 rounds of meiosis to produce a small cluster of cells, with each cell possessing 2 unequal-sized nuclei. The Polypodium hydriforme will carry these binucleated pseudosperm in a lidded pouch in the epidermis, which it then deposits onto pre-larval fish. So this tiny Cnidarian plants its gamete on the host fish, like a cop putting a tracking device on a suspect's car. And then, somehow, this binucleated gamete makes its way into the fish's reproductive tract and colonizes the egg cells, where it will live for the next few years of its life. In the fish ovary, within the fish's own egg cells, the binucleate Cnidarian cell will transform into an embryo complex, including a protective pseudo-amnion-like layer, and of course the embryo itself, which develops into an inside-out larva. Yeah, crazy, right? This is already insane, but it gets even wilder. Once embryonic development achieves the larval form, it almost immediately begins to replicate. Its body undergoes branching and budding to form a lumpy mass of multiple individuals. When this stolon-like structure has reached a critical mass, it will evert, or flip itself right side out, revealing the squirming nest of tentacles from dozens of clonal individuals. These newly revealed tentacles will help the stolon absorb yolk fluid in the fish egg, and when they're plump enough, they'll break out of the egg and split apart into free-swimming individuals. They'll mature on their own, and eventually reproduce through fragmentation, or that weird kind of asexual but pretending to be sexual replication. So these Polypodium hydriforme creatures are definitely a super creepy, super weird, obscure Cnidarian offshoot. They're also closely related to the Myxozoa, another obscure but relatively large branch of Cnidarian, with over 2,000 known species. The adult Myxozoan looks either like a worm or somewhat like a tiny transparent starfish with a few long slender tentacles, almost like the shape of a banana peel or a living caltrop. They're universally small, really small, with arm spans measured in micrometers, which makes them some of the smallest animals in the world. Research with molecular clock analysis suggests that the ancestral Myxozoans diverged from their Medusazoan cousins around 600 million years ago, and since then they've settled into the ecological niche of obligate parasite. They've evolved an extremely reduced body form. Almost everything they didn't need, they got rid of, be it tissues, pseudo-organs, even genes. They don't necessarily need to swim around in the water column, so they lost all their cilia. If nutrients can be absorbed directly, you don't need a gut, and lo and behold, they've evolved to lose their guts. Their nervous tissues have largely disappeared, and their epithelial tissue has simplified, with fewer glands and specialized structures. Many species, but not all of them, have also lost their muscle tissue. To move around inside the body of their host, they've developed a few amoeba-like strategies. Some of them will extend out filaments of cytoplasm that probe and explore the area immediately ahead of them, with the visceral mass being pulled along in the direction where it senses something that it wants, like nutrients, or an area with better oxygenation or something. On that note, not all of them even breathe oxygen. In a very recent discovery, it was noted that species Cnidaria salminicola has lost its mitochondria. It doesn't have a mitochondrial genome anymore, because the entire endosymbiont isn't there. It's gone. Cnidaria salminicola is thus the only known animal that can't breathe oxygen. To deepen the mystery, we currently don't know what it uses for cellular respiration. Like maybe it uses sulfur or something, we don't know. And for that matter, we don't know how it does it either. Now, these aren't the only genes they've lost. Evolution has also taken from the myxozoans many of the genes involved in embryogenesis and development, the genes involved in cellular communication and in multicellularity. I suppose some of this makes sense when they've evolved to be so small that they're basically extremely tiny multicellular creatures, made up of only a handful of cells. And this is only if they've retained multicellularity in the first place, as many species have been so reduced that they are now just a single cell. If this wasn't enough, evolution has also taken from them their gametes. Instead, they reproduce through multicellular spores, which are not totally dissimilar from the gemmules that we saw in the sponges. One thing that evolution has not taken from the myxozoans are their cnidocytes, those characteristic stinging cells that are shared among all of the cnidarians. The myxozoans still possess cnidocytes, they're just smaller and less powerful. Like the Polypodium hydroforme, the myxozoans also parasitize fish, but their life cycle is more complex and involves not one, but two distinct hosts. There's a vertebrate host of some kind, usually a fish, but a few species of myxozoan have adapted to parasitize other types of vertebrate animal, like amphibians, which you might expect, but also amniotes, like mammals and birds. The second animal host is usually some sort of worm, like a bryozoan ectoproct, or an annelid, like an earthworm. A small fraction of all known myxozoan species have had their life cycle studied in full, and a general pattern has become apparent. Let's start with the multicellular spore, called a myxospore, which is usually about 10 to 30 micrometers in diameter, but it can be much larger. The spore structure is typically some sort of sealed orb, with a mantle or a cortex of valve cells, and sporoplasm structures in the core or the center. Sprinkled throughout this envelope of valve cells are specialized cells that form extrudable capsules. The purpose of these surface-bound, capsule-extruding cells is to act like a primitive sensory system. They can feel out the external environment and determine if the spore is in the right place to enter the next stage of its life cycle. This right place is usually the belly of an annelid worm or a bryozoan ectoproct. In this environment, the capsule cells will extrude and anchor the spore in place along the gut lining. The valve cells facing the gut wall will open up, creating a crevice through which the sporoplasms can enter the intestinal lining. Here, the sporoplasm will embed itself in the host's gut cells, where it can readily absorb nutrients and grow larger. It will eventually produce around 8 smaller spore structures called actinospores. And by this point, the whole thing has grown into a swollen cyst in the intestinal epithelial tissue. When the time is right, the cyst is blown up, and these smaller actinospores are released into the water. This is where the myxozoans await their second host. In the water column, these smaller actinospores become part of the plankton, getting pushed around by the current and possibly absorbed or consumed by some larger creature. The spores eventually come into contact with a fish, or some other hapless invertebrate, with large numbers of spores attaching to their skin, to their fins or gills. Here, the spores will once again unleash their sporoplasms to invade the tissue and embed its visceral mass inside the host cells. In the epithelial tissues of this larger host, the visceral mass will grow into the larger adult form. The adult myxozoan maintains the ability to move through and between the host cells. For example, in many species, such as the Myxobolus cerebralis parasite of the rainbow trout Oncorhynchus mycus, the adult life stage is spent replicating more adult forms, while also migrating deeper into the dermal tissue to find blood vessels, or peripheral nerves. The myxozoan will follow these blood vessels, or these nerves, up to the trout's head, where they settle in the cartilage of the skull and the face. Once the Myxobolus cerebralis has situated itself in the cartilage matrix of the head, they'll produce myxospores. These are released from the orifices in the head, where they eventually find themselves in the bellies of annelid worms or various bryozoans, and the cycle begins again. The final group of strange cnidarians that I'd like to explore are weird basal creatures that may not even be cnidarians at all. Their placement is really uncertain, although more recent research with DNA suggests a close relationship with Cnidaria among all of the other early metazoans. These are the Placozoans, and the reason placing the Placozoans has been so tricky is because they're so unlike other cnidarians, and pretty much all other animals, pretty much everything else for that matter. Generally speaking, Placozoans are simple blobs of cells, about 1 to 1.5 millimeters in diameter. There's no consistent body plan, there's no consistent skeletal structure, and there's no organs. Awarded science journalist and editor for Science and Science News, Elizabeth Pennisi, described them as the simplest animals on Earth in an article called The Simplest of Slumbers. The former editor of Cosmos, Andrew Masterson, has described Placozoans as being, quote, as close as it is possible to get to being simply a little living blob, unquote. Regarding Placozoan anatomy, there is no mouth, there's no gut, and there's no orifices. To eat, small bits of detritus, or tiny, slow-moving organisms, are simply engulfed and absorbed. Reproduction is largely asexual, accomplished through simple budding, where the offspring starts out as a lump growing out of the side of the parent, and eventually just detaches and swims off. There are no appendages, there's no muscle cells, and no nerve cells. At most, there are a few cilia on the outer dorsal layer of flat epithelial cells that help the Placozoan navigate in the water. But even this outer layer of epithelia is loose, without a definite shape, like an empty bag submerged in water, or like a wrinkly circle of thick felt. It has a top and a bottom, but these are almost impossible to distinguish visually. Within the body, there's a small selection of cell types that work together to keep the little blob alive. Along the bottom, or the ventral side of the bottom, the epithelial layer is composed of column-shaped cells that are packed tightly together with their axes perpendicular to the ventral body surface. These too are equipped with cilia to swim or crawl along a surface, but among them are also specialized gland cells that secrete digestive products, and lipophil cells that can break down lipids and use the fatty acid molecules to build membranes. The Placozoan can absorb food directly through this lower surface, with help from the gland cells and lipophil cells. And throughout the body, in all layers, there are cells that release peptides, hormones, and mucus. They're also known to produce various venoms and toxic compounds, which are kept in vesicles in cells along their dorsal surface, which can discourage any inquisitive predators. Along the edges of the body, there are crystal cells that contain flakes of aragonite. These are presumably used to determine and maintain their orientation, in much the same way that jellyfish use statocysts. In the middle layer of the body, between the dorsal and ventral epithelia, there's a higher density of cells, particularly of fiber cells, which contain large vacuoles for storing nutrients and precipitates, and extensive populations of mitochondria. These fiber cells form a continuous, syncytium-like network that helps connect the epithelial layers and provides some cohesion to the body, and facilitates diffusion and communication between all of the epithelial cells. They also occasionally host algae and bacteria, forming symbioses that help the Placozoan to acquire certain nutrients. The Placozoans were once thought to be extremely simple animals that just never developed much, but more recently, they're thought to be the product of an evolutionary trend towards extreme simplification, as they lack almost everything that we see in other animals, except for the bare minimum necessary to even still qualify as a multicellular animal. But tantalizing clues left behind in the genome and the biochemistry strongly imply that they came from larger, more complex ancestors that were perhaps akin to a coral polyp. For example, their genome is much larger than necessary, with many genes being disabled or degraded. It shares close resemblance to the genome of Cnidarians, and for the last decade or so, all genetic research that has included them has placed them in close proximity to other Cnidarians, sometimes leaning a bit closer to bilaterians, sometimes a bit closer to ctenophores, but the link to Cnidarians seems consistent and robust. The Placozoans once had a true gut and a more consistent body form with basement membranes, but both of these traits were lost. They once had muscle and nerve cells, but these too were lost. Today, the Placozoans are almost microscopic blobs, descended from some tiny coral polyp-like creature or jellyfish ancestor. They have stripped away almost everything that it means to be an animal, including a mouth and a gut. But they have kept one thing, one trait, handed down from their Cnidarian ancestors. These are the acetylcholine blockers and metalloproteinases and all of the various venoms that they use to stun and paralyze their prey. They possess these neurotoxins in abundance, and they use them to stay alive and compete in an ocean full of creatures that are bigger, faster, and stronger than them. They use their venoms and their stingers to fight back, to disable and destroy prey animals that are physically stronger and much more massive than them. They have a reputation among humans, and perhaps other animals too, for packing a punch far above their weight class. And this, perhaps, is the greatest evolutionary legacy of the Cnidarians. And with that, we come to the end of episode 101. This was a long episode, and trust me, I could have gone on longer. The Cnidarians are just such wickedly cool animals, and there's so much variety here, and so much to talk about. But now it's your turn to go out and read a book on jellyfish and learn even more. If you think this in-depth exploration of the Cnidarian lineage was worth $1 or $2, then check out the Patreon page. You can become a patron for as little as $1 or $2 a month. So for a show that comes out once a month, it's a great deal. And if you really like the show and you want to show more support, we have Patreon tiers of $3, $4, and even $5 a month. Whatever you choose, however you support the show, it really helps by paying for web hosting fees and equipment and all of that fun stuff. So check it out, check out the official store, hit that like button, hit the subscribe button so you can see new biology content right when I post it, and as always, thank you so much for listening.